Hi everyone, welcome to Science Education Hub. In this video, we are going to discuss about the basics of DNA. Let's start with a brief history of DNA. In 1869, a Friedrich Mischer, a Swiss physician and a biologist, discovered a substance within the nuclei of white blood cells that is called nuclein. This was the first step towards understanding DNA. Mischer isolated DNA from salmon sperm, which is laid to be foundation for future discoveries. If you see the glass vial, the white substance which is present in the glass vial is the salmon sperm DNA. In 1952, Rosalind Franklin, a brilliant scientist, took the famous photograph 51 through X-ray diffraction, which showed the DNA had a helical structure. Unfortunately, her contributions remained largely unrecognized during her lifetime. If you see the picture here, if you see the picture here, this is helical structure which has taken the it was a photograph of taken in X-ray diffraction XRD. In 1953, James Watson and Francis Crick proposed the double helix model of DNA, a groundbreaking discovery in field of molecular biology. They were able to confirm the helical structure of a DNA based on Franklin images. Their work, published in Nature in April 1953, earned the Nobel Prize in Physiology and Medicine in 1962. Unfortunately, Franklin was not included in the award. What is a DNA? Now, let's talk about DNA. The DNA stands for deoxyribonucleic acid. It's a genetic material which is found in all living organisms from bacteria to human beings. The DNA is structured like a twisted ladder, which is known as double helix. The structure was first discovered by Watson and Crick in 1953. If you see the structure, this is a phosphate backbone, and this is the nucleotides adenine and thymine, adenine, thymine, cytosine, and guanine, which are bound together with hydrogen bond. Adenine pairs with thymine with the two double bond, and guanine pairs with cytosine with three, three, three hydrogen bonds. Why it's called as the DNA? DNA is known as deoxyribonucleic acid. The deoxy refers to sugar in DNA deoxyribose. It's called deoxyribose because it's a ribose sugar missing one hydrogen atom. This makes the DNA more stable compared with RNA. If you see in the structure, usually at a second carbon position, there was a missing oxygen group. Due to this, it is known as deoxy. D means removal. Deoxyribose. In the ribose sugar, the oxygen atom is removed. So that's why it is called as deoxyribose. And the nuclei comes from the fact of DNA was discovered in the nucleus, and it play a vital role in heredity and genetic information. The acid refers to phosphate group which is present in a DNA backbone, which give acidic properties to the DNA. Next, what is a nucleotide? What is a nucleotide? A nucleotide is the basic building block of our DNA. It consists of three parts: a sugar molecule, you will see here the sugar molecule, and a phosphate group, and a nitrogen bases. The nitrogen bases are adenine, thymine, guanine, and cytosine. You see, the adenine pairs with thymine with a double bond of hydrogen, and guanine pairs with cytosine with a triple bond of hydrogen bond. What is a nucleoside? A nucleoside is similar to a nucleotide, but it contains only nitrogen bases and a 5 carbon sugar molecule, usually deoxyribose in DNA. If you check the nucleoside, the nucleoside which is only present sugar molecule and nitrogen base. There is no phosphate group in the nucleoside. If you add a phosphate in this, it is known as nucleotide, and if you remove the phosphate, it is known as nucleoside. Now let's take a closer look at the structure of DNA. The DNA molecule was a double helix structure where two strands of nucleotides run opposite direction. If you check the DNA, it runs 5 prime to 3 prime and 3 prime to 5 prime in this direction, and the bonds were formed like this. Thymine and adenine, in between this there is a two hydrogen bonds, and if you check guanine and cytosine, in between that there is a three hydrogen bonds. And next, if you look at the structure of our DNA, this is known as minor groove. It is known as minor groove, and this is called a major groove. Major groove and then a minor groove. If you check the dimensions of the DNA helix are quite fascinating. The diameter of the DNA is 2 nanometer, and one complete turn of the helix is 3.5 nanometer. If you check here, from one minor groove to another minor groove, one minor to another minor groove is known as a complete turn. It contains 10 nucleotide base 10 nucleotide bases, and the size of the one turn is 3.4 nanometer. This is the types of DNA. There are different types of DNA, such as bDNA, which is most commonly found in cells. There is also called as Z form and A form DNA. Usually in all living organisms, B form DNA is present, and it was a widely accepted model. And the question, a DNA molecule has 5,000 base pairs. A DNA molecule has 5,000 base pairs. What is the total length of this DNA molecule in nanometers? The DNA base pairs is 5,000 base pairs length. If it is a 5,000 base pairs length, what would be the length given a nanometer? If it is 170, 1700, 3400, or 500 nanometers? Please drop your answer in a comment section. Thank you for watching. Please do like, share, subscribe. my channel for more videos. Thank you.